Today I will be telling you about the research we've done over the last 10 years where we have isolated this strain of Lactobacillus johnsonii, and we are very interested in trying to understand how this strain can prevent autoimmunity. So first I will tell you a little bit about diabetes type 1. It's not your regular diabetes that most people get. This is the one that you have a genetic predisposition, you have genetic markers that are associated with the disease, and is mediated by the immune attack to the beta cells in the pancreas, and then they stop producing insulin. Diabetes type 1 affects a large number of Americans, but it's mostly the onset is during childhood. And so this disease is increasing really fast. And if we look at this figure, if we look at the rate of progression, mostly in Finland, where we see a bigger acceleration on the disease, but also in the United States, this rapid increase in the disease onset cannot only be explained by the population growth. So there is several environmental factors to the onset of this disease. So many years ago, Dr. Atkinson put this figure out, and I think it's still viable today. And it's where we know that about how beta cells are recognized by T cells and how cytokines induce apoptosis of these cells by the linkage with T cells. But then after many years, what we now have understand very well, and most recently has just been published in Nature, the microbiome of type 1 diabetes, is that an altered microbiome together with some will promote a leaky gut in the intestine, and then all these antigens from microbiota, but also some other dietary antigens will induce the activation of antigen-presenting cells that will then recruit T cells and make B cells to produce even more cytokines that will technically, what is thought as the mimicry theory is that originally these antibodies are directed to some antigen in food or microbial origin. And then once this is exterminated, then they can target beta cells. This is the mimicry theory, but we have no proof of that so far. So we had a very simple idea, and this was, let's analyze in animal models what happens when we have a rat that is genetically predisposed to get diabetes, that are these diabetes prone rats, and compare them to the ones that don't develop, and look at the microbiome. And if you some of you are old enough, you know these are ERISA profiles, and this was when the microbiome studies were just started. But early on, we were able to identify that lactobacillus were correlated with the disease resistance there. And this is data that we later on correlated with sequencing by 454 and Illumina. But what I did was to isolate this lactic acid bacteria and then test them in the same model. Very simple idea. So what we did was to get these diabetes-prone animals and then start the administration of lactobacillus either as soon as they were born or post-weaning when they start on solid food. And then the good thing about this model is that they develop diabetes. It's very stringent between the day 69 and 110. If they don't get sick by then, they will not get sick. Very simple. So the strains that I isolated were hundreds, and then I end up choosing two that were a Lactobacillus johnsonii and a Lactobacillus roteri. The basic idea was that these are strains that species that you usually find in humans. We didn't care about the ones that we only find in animals. So what we found was that in here you have in blue Lactobacillus johnsonii and in red Lactobacillus roteri and in black the controls, is that if we start as soon as they are born, the rats, we didn't see any difference in the progression of diabetes. But then if we started after, when they were on solid foods, then we see a significant difference on the Lactobacillus johnsonii group. Well, the other two got, actually Lactobacillus roteri seemed to accelerate disease progression somehow. So from here, we started doing a lot of mechanistic studies. We wanted to understand how the bacteria is doing this awesome effect that we we're not very clear, but the first thing we started looking was in the ileum, and we observed important differences on oxidative stress markers. As many of you know, these are related to inflammation in the gut. And most of these markers were related to the health status of the animals. So here in this line, you have Lactobacillus johnsonii, and everything that goes up means they are inducing disease in animals that have the disease. In this case, for example, for these two markers of oxidative stress like catalase and glutathione reductase, they were only related to the health status, but then all the other markers were really related to the administration of Lactobacillus johnsonii. So we have an excellent marker there already. But then what also was interesting, the reduction in the expression of inos, that here in the control group seems activated, but then with Western blot we prove that is related to a health status. And this is an important marker because this is what the beta cells, that's the production of nitric oxide is what kills them. So moving on, we look at other regular markers like TNF-alpha and interferon gamma, and we did see a very strong reduction by the Lactobacillus johnsonii administration. In the mesenteric nodes, we also noted important differences, but not in the markers that we expected. Most people usually report anti-inflammatory roles for this kind of group of bacteria. In our case, we observed there's a very distinct Th17 bias that was induced, that was related to a health status. And back in the day, it was very complicated to explain this, and most people were not on board with this. But then since this study was published, other research groups have found that the induction of this Th17 subset is what actually prevents diabetes, also in the NOD model. So we were really wondering, what is this bacteria doing, and where can we start to look for systemic markers, because you're giving the bacteria orally on the intestine and it's inducing all these markers systemically. So we thought about an enzyme that is the one that catalyzed the first step in the conversion from tryptophan to kynurenine. And this important enzyme is found in many tissues, is ubiquitous to the intestine too, and is highly expressed. And these ratios of kynurenine to tryptophan is what is gonna determine which kind of T cells you're gonna have circulated, either regulatory T cells, or you can induce changes between Th1 and Th17s. So we look at this enzyme's first expression in the ileum, and here you have the levels on the control groups, the ones that got diabetic, and then on the Lactobacillus johnsonii group. So this bacteria was reducing the expression, and we observed, and this reduction in expression was also correlated with the levels of kynurenine that were much higher in the diabetic rats, but then lower in the Lactobacillus johnsonii group. So after this study, we did a lot of in vitro experiments. We were still wondering if the bacteria will only affect expression through maybe interferon gamma reduction, or if we were also reducing activity, since we have very strong values on the plasma of these metabolites. So we got a clone of the IDO, the human IDO. We purified the enzyme, and we really start putting a lot of supernatants from this bacteria to see what it was doing. And what we did find was that, in fact, this bacteria is making a lot of hydrogen peroxide in these preparations, as you can see in this figure. And this, if we look at IDO activity in vitro, then the addition of hydrogen peroxide was really inactivating the enzyme, and then this effect could be counteracted by catalase. So the same effect was observed. And what we are adding here, I forgot to mention, is we are already adding the supernatant from Lactobacillus johnsonii. So the treatment with catalase will really inhibit the enzyme back. What was okay, is the bacteria producing hydrogen peroxide in the intestine? And the answer is yes. If we look at all the different portions of the gastrointestinal tract, this is the group that was fed with Lactobacillus johnsonii, the dark blue. We observe a significant difference in the ileum, and this is what we know by culturing experiment, that is where the bacteria is colonizing in animals. The other possibility is, if you are making lower kynurenine, is that you are directing the pathway towards the production of serotonin. And this is very important because this can have systemic effect and or can also severely affect the brain-gut connection. So we look first at the production of serotonin in the ileum, and we observed a significant difference on the Lactobacillus fed group, and this effect was also found in peripheral blood serum. So then we wonder, is it getting to the brain? And then are we having the same effect in the brain? Because it has been seen that modifications of the serotonin, it can travel directly to the brain and can pass into the central nervous system, having effects on behavior or feeding patterns. So we determined by Western blots the presence of the enzyme in the brain, and we observed that in the Lactobacillus johnsonii group, the expression levels were really decreased. So how we transfer all this to human studies? Because here we're not very interested on curing the diabetes in animals. We want to cure or prevent diabetes in humans. So having proved the concept that we can isolate a member of the microbiome and prevent disease in animals, what we wanted to do is we want to really get into humans and do this. So our first stage, that is the proof of concept, has been completed. So we ran a clinical trial or a safety trial with these strains in humans. And this, that's what I'm going to show you briefly now. We ran this study. It was completed by a baseline. We gave them patients capsules with Lactobacillus johnsonii for 8 weeks, and then followed by 4 weeks washout. And the placebo here was dry milk. That's what how the bacteria was prepared. And then we did a lot of safety parameters, as FDA likes, a lot of daily questionnaires, weekly questionnaires, and in blood samples we look at regular safety parameters. But we want to look at mechanisms, so we did a very strong immunophenotyping and, of course, metabolomics focused on tryptophan metabolites. And in stool samples, we look at transit survival and microbiome. So the first thing that was really interesting for us when we look at the symptoms is that we saw a very strong effect on the gastrointestinal distress syndromes, like a strong decrease in bloating, flatulence that started a week after we started providing the probiotic. And then it lasted throughout, even after the washout period. We also saw important differences on the epidermal syndrome, like itching, skin rash, also on cephalic syndromes, so a decrease in headaches, dizziness, and also psychology, we saw strong differences on anxiety. So people taking this probiotic it was really strong. So by seeing this, we look at what is the results or how the metabolites will explain that. Remember, I told you this is supposed to decrease your kynurenine levels at expense of increasing serotonin. And what we did metabolomics, and I'm just marking how we went into serotonin and all the derivatives of kynurenine. Today, I'm only going to show you the results on kynurenine, tryptophan, and the KT ratio, that this is the standard way of showing IDO activity. And what we observed was significant differences between the placebo and the control, but only on a subset of the population. So if we did a stratification by levels of lactic acid bacteria, and in the group or in the subjects that they were increasing over time the amount of Lactobacillus johnsonii, those were the ones that show a strong correlation with a decrease in kynurenines, on increasing tryptophan. And it was also reflected in the kynurenine to tryptophan ratio. So this gives us an idea of how we can choose our responders from non-responders to treatment. So we did also immunophenotyping, and I'm not going to go through this very complex figure, but I just want to mention that we divided the blood samples in 6 panels, so we could have a very deep analysis of the immune responses at each time point. And I'm only going to show you results where we saw very strong differences, starting by monocytes and natural killer cells. As you can see here, monocytes increase over time on the Lactobacillus johnsonii group, as well as natural killer cells. And when we look in this subgroup, which ones were really increased were the activated natural killer cells. We also observed that the concentration or the percentage of CD4 positive T cells were decreased in the Lactobacillus johnsonii group, but there were not main differences on their activation status. Looking at T effector cells, and these are cells that, remember I showed you in animals that we have differences on the Th17 subsets. Here we observe, again, that we have differences mostly on the group Th1, Th17s, and these are T cells that are not very well studied. They just give you positive markers for both. So we don't really know the significance of this yet. But if we look here, on the activation status is mostly what changes. So Th1 cells and Th1, Th17s are highly activated after 8 weeks and continue to be after 12 weeks of treatment. By looking at other markers that may explain these shifts in immune cells, we observe no differences on interferon gamma or TNF-alpha or other markers. But you also have to remember these are people that is healthy. So many of the measurements were under the detection limits. But we did see an increase in IgA after 12 weeks. So just to summarize this study, we observed that this preparation, as expected, was safe and produced no side effects on the subjects. We did not see any shifts in the microbiota, and I didn't went through that because of the time, but with the microbiome in all these subjects was so variable that it was not possible to see shifts induced by lactobacillus, although we see a non-significant increase in lactic acid bacteria over time. We did see significant differences in the gastrointestinal scores, and many of the cephalic syndromes and epidermal symptoms can be explained by this shift on Th1 responses. You know, in allergies, you have a shift from Th1 to Th2. We also observed that lactobacillus can modulate the concentration of metabolites of the tryptophan pathways. And we observe and we identify markers for the mechanism of Lactobacillus johnsonii, mostly on monocytes, natural killer cells, Th1 cells, and I didn't go over this, but we did see a strong increase on cytotoxic T cells, CD8 positive T cells. So having done the proof of concept phase 1, we are now getting ready for our phase 2 trial, and in this case, it's gonna be with kids with diabetes type 1, again, proving safety before we can go into a preventive phase 3 trial. And I want to end with this slide that is very complex, but the interest in my lab is really to focus on mechanisms. So we are trying to do different mechanistic studies to really understand the magnitude of the pleiotropic effects that we see. On animals, we have looked at the tryptophan pathway, that is what I talked today, and we have recently published 2 other papers focusing on how Lactobacillus johnsonii can decrease the activation of the inflammasome by inhibiting the maturation of caspase-1. And this is also impressed that where we look how Lactobacillus johnsonii can modulate the mTOR pathways. In humans, what we are mostly interested in is trying to understand how the bacteria in the intestine, again, is sending components. We don't think the bacteria is migrating to the pancreas, so some components of the bacteria is going from the intestine to the pancreas and then to the beta cells. So we are currently doing studies with beta cells to study but different bacterial components to identify how the bacteria is helping them to remain in a healthy status and not switching to T-cell infiltration and death. So of course, this is a teamwork too, and many members of my lab were involved in this. Guillermo was a postdoc in my lab who did most of the human trial together with Amanda Ford in Dr. Dahl's lab. Dr. Gonzalez's lab and his grad students have done most of the mechanistic studies that I presented. And of course, we have all the diabetes type 1 group where we do most of the immunophenotyping. With them, they have trained my students on this. Todd, who really gave us insights on all the T-cell maturation, and of course, Timothy Garrett for the metabolomics, and Eric Triplett for the initial microbiome studies. And these studies were funded by JDRF and NIH and some seed funds.